All right, in this video I want to go over some of the stages of reproduction and development in animals. We're going to talk about some common things that occur in all animals and some specific things with humans especially. So this is a human embryo. That's kind of what we look like. This is at day 49, which is kind of interesting. Not even 2 months gestation. Embryo's about 30 millimeters, which is about an inch long. It's kind of cool. So we're going to talk about some things. All sexually reproducing animals show a similar pattern of development from egg to adulthood. So one of the things to remember, some organisms, they undergo what's called metamorphosis. And some development happens inside of a uterus. No matter what, though, you see some similar patterns. The first stage is fertilization. Once the sperm reaches the egg during fertilization, the head of the sperm actually releases some hydrolytic enzymes that will allow it to penetrate the egg. Once inside the egg, there's a signal from the sperm to some receptors within the egg that change the actual surface of the egg, that keep it from allowing any more sperm in. And that ensures that only one sperm fertilizes the egg. There's a little chemical reaction that happens. In some species, though, this response can be elicited without a sperm, and these species can undergo what's called parthenogenesis. In parthenogenesis, you can get offspring that are not quite sexual reproduction. It's producing offspring from just one adult. The adult that's formed through this process, because we're just dealing with an egg, is a haploid individual. Okay, so it's just one set of chromosomes, not two. An example of this are honeybees. Honeybee females will undergo parthenogenesis due to signals that cause this process to happen and the egg to begin developing into an embryo. And through this process, parthenogenesis, they generate the drones. And the drones of the honeybee hive, from what I understand, are the ones that are providing the sexual reproduction with the female. So because they're haploid, they're just generating the sperm cells that are going to join with the eggs of the female. So they aren't the workers. The workers are diploid, but the drones are just haploid. Following fertilization, the fertilized egg, which we now call a zygote, will follow a prescribed pattern of development. And there are 3 stages to this pattern. We have cleavage, gastrulation, and then finally differentiation. During cleavage, there is rapid division of cells. So mitosis occurs rapidly, and while this is going on, the embryo or the zygote does not change size. So it keeps dividing, and each cell is progressively smaller, so that by the time it's finished, there's a ball of cells that are the same size basically as the original zygote. And this ball of cells is called a blastula, or sometimes called a blastocyst, and it surrounds a fluid-filled cavity called a blastocele. Most of this process is not controlled by the genes of the embryo, oddly enough, but by mRNAs and proteins that come from the original maternal egg cell. So here's kind of what that looks like, a little drawing. Here's the exterior of the blastula, and then this would be what it looks like on the inside. We have this fluid-filled cavity here called the blastocele. Here are some actual images of cleavage occurring in a sea urchin. We start here with an egg. Here is the zygote, the single-celled fertilized egg, and then we have a succession of the cell divisions, mitotic divisions, and eventually ending up with the blastocyst or blastula that has a fluid-filled cavity on the inside and then a ball of cells on the outside. Here's another example. This is some images from cleavage in a frog embryo. You see the process is very similar. So despite the fact that sea urchins and frogs are very, very different, and even humans very, very different, a similar process occurs. Following the formation of the blastula, cleavage, the embryo will undergo what's called gastrulation. Gastrulation is the process by which the embryonic cells begin to become organized. And the way that gastrulation progresses determines the overall pattern in the adult. So you can see species by species there is some difference in the process of gastrulation, although it is very similar. During the process, one wall of the blastula will begin folding inward. This is called invagination. It will fold it. It will form an invagination. It will fold inward, and you can kind of see that happening here in this image. An opening will form, which will become the anus. So it's the backside that forms first in animals, and this is called the blastopore. So you can see it here on this end. The end product of gastrulation we call a gastrula. So here we can compare the blastula and the gastrula. So what's happened here? This fluid-filled cavity, now the edges of the blastula have begun folding in, and here you can see they're beginning to have more of a structure to them. By the end of gastrulation, there are three distinct what we call germ layers. These are the early layers that are going to become the different tissues of the adult animal. We have the endoderm, the inside layer, which will become the internal organs, such as stomach, small intestine, all of that. We have the mesoderm, meso means middle, this is the middle layer, which will eventually become the bones, the blood vessels, the muscles, and the connective tissue within the body. And finally, we have the ectoderm, which is this outer layer. You can see it here in blue. The ectoderm will become the skin and also the brain and the sense organs, the nervous system. Here are some images from a sea urchin, again, I believe. We have the fertilized egg, the zygote. We have here the formation of the blastula. You can see the fluid-filled cavity. And then finally, these images here are showing the different stages of gastrulation. So I believe this is our gastrula with the early germ layers forming. And then here's the final embryo of the sea urchin. Following gastrulation, we have a process called differentiation that occurs, and during this time, the embryo's tissues and organs form, and they begin to differentiate and become the different tissues and organs they're going to be. This is due to expression of different genes in the cells. For instance, the lens of the eye, shown here, produces a special protein called crystallin. Crystallin, it looks like I spelled that wrong, there's no E on the end. Crystallin is a protein that's transparent. So it's very, very unusual. It's only expressed here in the lens of the eye. And the gene for crystallin is present in all your cells. So your heart cells have the gene for that. Your skin cells have it. The cells in your big toe have it. But they're not expressing crystallin. During differentiation, the cells that form the lens are the ones that turn that gene on and begin making the crystallin. Now, another process that's involved in differentiation, not just turning genes on and off, but how that happens, has to do with something called embryonic induction. This is when one group of cells sends signals that affect the development of nearby cells, and it has to do with release of local signal molecules called morphogens. And scientists have found that the concentration of morphogens actually are what influences what genes are activated and which are turned off. And I just have a little diagram here to help you understand that. As the concentration of morphogen decreases, we get differentiation of different kinds of cells. So for instance, here we have the source of the morphogen. The morphogen, it will be highest right near the source. So these first two cells become one type of cell because they're in high concentration of the morphogen signal molecule. The next three cells are in sort of this midway concentration of the morphogen molecule, and they become a different kind of cell, and then we have these last few that become a third kind of cell because they have very low levels of the morphogen. So depending on what cells we're talking about and what type of morphogen we're talking about, these signals can determine how these cells will develop. Another important process in development as the embryo is forming is apoptosis. And I know that you've read about apoptosis before, you've heard about it, but this programmed cell death, apoptosis, will actually help sculpt body parts. Very important. Cell death is very, very important process in the development of an organism. One example is the human hand. You may have seen images of human embryos early on. They sort of have this webbing between their fingers. Without the removal of this tissue that's in between the fingers, you don't get fully developed hands. And you may have heard of people having sort of webbing between their fingers. There are some disorders associated with that, and it's due to the fact that perhaps apoptosis didn't occur properly in between there. Another example is the loss of the tail. As a frog goes from being a tadpole to a fully adult frog, its body has to lose the tail, and it's through apoptosis, this programmed cell death, that the tail is removed. Another important thing that happens during the last differentiation stage is that cells migrate. As part of the development process, cells will migrate to different parts of the embryo to form distant tissues. So you can think about the ectoderm. The ectoderm is going to generate all the skin, but also the eyes, ears, nose, tongue, the brain, the spinal cord, all of this stuff. So it's going to be located in different parts of the embryo. So these cells that are going to become those tissues have to move. How they do that is they move along in an amoeba-like fashion, and they're anchored as they go to the membrane of another cell with adhering proteins. So you can see this here in this diagram. This cell is just kind of stretching and inching along here until finally it's moved that distance. Of course, we're talking about small distances because the embryo is very small, but the cells are even smaller, so it does have to go some distance. But what's going to happen is they're going to just kind of ooze along like an amoeba and get and anchor themselves like you would, like climbing up a mountain or something. All right, last little bit, I want to talk a little bit about the neural tube. An important stage in the development of a vertebrate is the formation of the brain and spinal cord. It happens by way of this structure called a neural tube. This is how the brain actually begins to form. It's regulated by morphogens that are found in the mesoderm cells in a structure called the notochord, which is a unique structure to all chordate animals. That's how they got their name. Notochord indicates it's a chordate. So cells in the notochord actually release morphogens that cause thickening, first of all, in the ectoderm. So here we see some thickening and some folding due to release of morphogens from the notochord, and that's going to form something called the neural plate. Eventually it will fold upward, forming what's called the neural groove, and finally the edges meet and form the neural tube. Now this is occurring all along the embryo, so you can see it kind of happening here. This is early in the formation of the neural tube, and this is later in the formation of the neural tube in the embryo. Now, this is interesting because, of course, it's the brain, and the brain's always interesting, but also it's a tube, and eventually the top end of the tube and the bottom end of the tube have to be closed off. And there are two important disorders that are associated with this not happening properly. If the bottom part of the tube, the caudal end, that's the tail end, doesn't close properly, then when the baby's born, it has a disorder called spina bifida. You may have heard of spina bifida. It's really difficult for families and children that have that disorder, but it's caused by not completely closing the tail end of the neural tube. If the head end or the cranial end of the neural tube doesn't close properly, there's another disorder that we see, and that's called hydrocephaly. Hydrocephaly is a disorder where the neural tube doesn't quite close, and they used to call it water on the brain because hydrocephaly, that's what that means. But the brain, the upper part of the brain is not completely enclosed, and fluid builds up, and it can cause brain damage. Now for both of those, we have treatments available, and for hydrocephaly, usually there might be some brain damage, but not a lot these days. Spina bifida, I'm not sure exactly how they treat that, but I know they have treatment for it. So there's an overview of development in vertebrates, and that's all for this one.